Hi everyone! This is the first of 3 videos that talk about the various aspects of protein synthesis. And by this time, you should have noticed that this particular unit is very heavy in Skill A, which is models and representations. Everything that we know about DNA and protein synthesis and replication is all based on some very sophisticated investigations in cellular biology and chemistry. In order to understand how all of those investigations come together, we actually use models to show what the thinking is of the people who are interpreting the results. When Watson and Crick determined what the structure of DNA was, that structure actually also indicated to them the ways in which DNA did its 2 jobs, which is replication for one, and second, to hold the information for making proteins. Models and diagrams actually help us all to communicate about what we believe is going on and make it a little bit easier for us to discuss. The diagram you see here is actually demonstrating the entire process of protein synthesis, and by the time we finish all 3 of the videos, you will understand what all of this means. This first diagram shows us transcription, which is a process that happens in the nucleus. To get to the code, helicase unwinds and unzips the double helix. The purpose in transcription is to copy the code for one section of the DNA, called the gene. Because there are letters on both sides of the ladder, one of those sides does not contain the information from the gene. This is called the coding strand. The strand that has the gene is called the transcription strand. Because the DNA can't leave the nucleus, we need to make a copy of the gene. So an enzyme called RNA polymerase is going to land on the open DNA right about here in an area called the initiator region. And what it's going to do is it's going to take these structures, which are floating around the nucleus. These are free nucleotides, and they happen to be free RNA nucleotides because they're going to be making a structure called mRNA. RNA polymerase will be using the DNA as a template as it follows the complementary base pairing rules, putting the RNA nucleotides into the DNA template. As it does that, it is going to be making this structure here, which is the mRNA. It will continue to move down the length of the DNA until it gets to the end of the gene. We can see the RNA polymerase is represented by this large gray circle that's in the back of the DNA. Once the RNA polymerase gets to the end of the gene, the mRNA is complete, and it is going to detach from the DNA template, and it's going to carry the gene out of the nucleus so it can go to the cytoplasm where it can be translated.